half a minute, 45 seconds. Talk to your neighbor about this question. If you were going to have some sort of a color code that represents the nucleotide-nucleotide bond, so A to T, as that particular sequence of 4 colors, and T to A in a different sequence, which of these codes, the panel on the left or the panel on the right, would you rather have if you were a protein, you were trying to figure out what DNA sequence you're finding? What do you think? So the problem with the right side, it's the left side wins. The problem with the right side is if you were a protein and you were looking at this sequence through this color code, which is not, of course, what happens. Proteins are colorblind. You wouldn't be able to distinguish as a protein an AT base pair from a TA base pair because the colors are the same. The pattern's the same. And the same problem you have for GC versus CG. So if you were a protein and you were reading the 5' to 3' direction and you saw red, green, red, that is green in the middle, right? Gray? Gray? Crap. Is it red? Yes. Okay, good. So if you saw red, gray, red, you wouldn't know if you were reading an AT base pair or a TA base pair. And the same with GC. You wouldn't know if you were looking at GC or a CG. So if you're a protein and you're trying to read the sequence of a DNA molecule from 5' to 3', A and T look the same to you using this code. That code, of course, is an abstraction, but it tells us exactly about what the proteins do when they're sensing, when they touch the DNA and they're sensing what the sequence is that they're looking at or they're interacting with. So what is the information, not color? What does color represent? What is the information that proteins can use to feel what sequence they're interacting with? It's the molecule of DNA. So what about the molecule of DNA?
Like the slight differences between adenine and thymine and stuff like that.
So it's subtle chemical differences between the 4 nucleotides. So those colors represent different bonds that can be made between the protein, electrical interactions, the way proteins feel, between the protein and the nucleotides. So if you look in the major groove, for example, of an AT base pair, then those colors represent different bonds that can be made, hydrogen bonds, electronegative, electropositive interactions between proteins and the edges of those base pairs. And each AT and GC differs in the order of what's red representing? Right, so red, there's a pair of electrons there on nitrogen. This is why you have to take organic chemistry for genetics. There's a pair of electrons there, so that's an electron donor. Blue, there's a hydrogen that can form a hydrogen bond with a protein that's touching that piece of DNA. What about oxygen? It's an electron donor as well. Right. So there are positive and negative charges along the edges of the nucleotides, and that's what proteins feel or interact with that makes proteins stick more to one DNA sequence than another. You do not have that image. You do now because it's in the recording. And likewise, now you have, oh dear. Let me try that again. That was ugly. I want to give you this image too. Because maybe it gives, I don't know, hopefully it's helpful to you. It gives a better sense of the differences between the edges of the nucleotides. So when you look at a DNA double helix straight on, you see the base pairs as flat objects. But if you pull one of those out and you flip it so you're looking at it from the top down, like this, then you can see that there's a difference in AT bases than GC bases on the edge. And that's the edge that a protein can interact with, because a protein's coming at the screen like this. It's going to bind to the DNA molecule. It doesn't feel the insides of the base pairs. It doesn't feel where the base pairs are hydrogen bonding with each other. That's in the middle of that double helix. The only thing that proteins can touch or sense is the edge of the double helix. So that's the information along the edges that proteins can use to sense what is the sequence of DNA that I'm actually physically touching. And it turns out, I didn't point this out on the slide, I should have, a couple slides back, the major groove, the point of that whole color coding thing, the major groove has more information about the individual identities of the nucleotides than the minor groove. So the minor groove, the panel that was on the right where the colors looked the same if it was AT or TA, that's why most proteins do not interact with the minor groove of DNA because there's no information there that helps them figure out what DNA sequence they're interacting with. The major groove's where all the action's at. Get your major groove on.